Hi, I'm Jonah Comstock, editor-in-chief of pharmaphorum. I'm here at CAR TCR in Boston with Tim McGuire, director of business development at Resilient. Uh, First, thanks for coming and chatting with me.
It's my pleasure.
So Resilient is a CDMO in the cell therapy space, right?
That's correct. And we cover gene therapy and nucleic acids as well as biologics and vaccines too.
Okay.
Yeah.
Yeah, I saw two of your team members on stage earlier, talking a little bit about what you do. But tell the folks. I mean, there's a lot of manufacturing organizations here. Obviously, this is kind of a exploding space. What's your value pitch for as to kind of what sets you apart in this space?
Sure. I think, while we bring novel platforms and gold standards for cell and gene therapy, where we try to differentiate ourselves is in three areas. So one is we have extensive partnerships with clinical organizations such as MD Anderson, where we have one of our joint ventures that helps us get to first in human faster. Secondarily, we work with clients across the full spectrum. So as early as early stage academic spinouts all the way through large pharma and biotech. And with that, we're able to, or because of that, I should say, we enable different business models, especially in this environment where it's hard to raise venture capital. So we either do aligned incentives so that, again, companies can remain lean, but then we also have strong partnerships with granting agencies such as CIRM and Secret to help those startup companies or even mid-cap companies that are looking to finance new clinical trials have non-dilutive capital. So we try to, start all the way from financing all the way through the process development and manufacturing with our clients.
Oh,
So
that's super interesting. So you're really helping to kind of foster this environment of startups and innovation in this space.
exactly, we want to see the medicines actually, the therapies actually get out to the patients. And we work with a lot of rare disease and ultra rare disease companies as well, or I should say nonprofits as well too, to help bring their therapies to market. Yeah.
Is it like a pipeline approach where like you work with people on their clinical and then they have a good experience and they come to you for commercial and then that's the big business? Or?
Exactly. Or and people even take one-offs too. Some people come to us just for regulatory support. Some people come just for analytical, and then we even have advanced systems that we see, kind of in the future. So the early access partners, for instance, we have a platform that reduces turnaround time for testing for cell therapy. So with the patient, so the actual cell therapy can get to the patient quicker. So that's our kind of not moonshot projects, but 3 to 5 years out. But with, like you said, with those, trying to bring, working with our clients in different ways, right? So not only manufacturing, but through future-looking offerings as well as the regulatory and clinical support too. Yeah.
There's been so much conversation, and when it comes to the manufacturing side, about just getting ready for this flood of that's going to come when all these products get approved and need to be manufactured at scale, and all these pieces of like making sure that there are centers of excellence to do the actual um, administering the therapy, but really just like when it comes to manufacturing, that turnaround time, that quality and reliability, all those things, you really have to kind of push them to the limit if this is going to be a viable, successful therapy for patients. So, what are some of the ways you guys are kind of innovating, and what are some of the metrics and KPIs you guys are thinking about when you're sort of trying to really perfect these manufacturing processes?
That's a great question. Yeah, I think so. If you look, we'll just pick on cell therapy kind of, across the field, even though we have a lot of viral vector as a drug substance that goes into the product. But I think, the biggest things with our CAR T, CAR NK, trying to reduce that 14-day, manufacturing time down to about a week. And then, like I'd mentioned before, with so release testing really is a sticking point because that can take 10 to 15 days, and we're looking for a 24 to 48 hour window that we bring that down to. And the major KPIs with the manufacturing, it's really kind of the quality of cells that come in, but then being able to expand them rapidly so that you can get it into the patient again, because they're typically quite sick, so you want to be able to get that. And like I said, with the release testing, being able to move away from the traditional methods and look for more high throughput like sequencing methods or a variety of other assays that cut that down too, so it can get into that patient faster. Yeah.
So one of the things I've heard a lot at the show is about allo versus auto,
Sure.
and this sense that the industry is going to move more towards allogeneic because it's more scalable and it's more off the shelf, as people say. So where does that look? How does that look from your perspective? I'm sure you work with both, but are you gearing up for kind of a shift in those ratios? Or?
I think, it's, if we look right now, they're mostly autologous, right? Just because there are the challenges of the allogeneic. So I think the first wave, at least for the next 5 to 10 years, will probably be large manufacturing setups with, closed systems before we can move over to that bankable kind of allogeneic cell therapy. So depending on the sites we have and the client needs, we try to fit a process as either auto or allo into a manufacturing site that can handle it at commercial scale where you have a lot of patients coming through. There's certain cell therapies like TIL, for instance, tumor infiltrating leukocytes, that will always be auto though because it comes from a patient biopsy. So we try to fit the process, unit ops, the workflow to a site that can handle those large, the maximum amount of patients that you'd see on an annual basis. But yeah, for sure. I mean, if we can get to the point where it is allogeneic, it's frozen, and you can kind of ship it anywhere in the world, you'll definitely see smaller footprints and much quicker delivery to the patients. Yeah.
What else have you been seeing or hearing at the show in terms of trends that are interesting, things you're excited about? What are you going to go home and be talking to your team about?
Sure. No, I think just the newer cell types. So I've been seeing a lot of great presentations here around. So kind of your iPSCs, again TILs as well, and then even hematopoietic stem cells are. There's a lot of buzz I see here. Maybe not at other conferences throughout the year. So going back and reading my peer-reviewed journal articles and talking to our subject matter experts about that, how we can keep augmenting those cell therapies as well too.
Yeah, it's been super interesting to me how different each presentation is from the others because there's such a world of kind of approaches to, for instance, the solid tumor problem, and the macrophage presentation this morning is very not like anything I'd heard. So
Yeah, yeah. It is. It is. With especially macrophage being able to create exosomes for delivery, that opens up a whole new world for cell delivery. Like you were saying, solid versus liquid tumor, the advancements they have, even in the CARs, being able, or just even in the antibody space, being able to have shielded antibodies now, so that you have direct site applicability and you don't have like full systemic
yeah,
cytokine release syndrome and things like that. So you're right. It's the nuances in the science that people have now, even with the tried and true modalities, is pretty mind blowing. Yeah.
how do you create standardized manufacturing for an industry where there's so little standardization that people are doing so many different variations on how to develop these therapies?
That's a great question. So I think we're lucky in that if you look again just at cell therapy, your hematopoietic stem cells, your CAR T, your CAR NK is pretty much the same unit ops or closed systems that you could use to manufacture those. It's really, I think, with the TIL, it's a bit different because you need two expansion phases. So our joint venture with MD Anderson down in Texas, that team has extensive experience in that, and they've been able to optimize that process. So you're right, that one's they've brought that down to a platform, but that was a multi-year effort. And if you look at regenerative medicine coming up now with iPSCs, that one I think is going to be more difficult to reduce to practice because it's really three separate phases. You need to reprogram your cells, and there's companies out there that are experts at that. You need to be able to expand them, and it uses different processes than we have now with, with respect to those closed unit ops. And then you have that back end differentiation, which is really unique to the end cell type you're going at. And I think that's the hardest part for iPSC to kind of standardize. Yeah. Well, again, we're lucky with at least CAR T, CAR NK, and HSC in that they essentially use the same processes.
Right.
Yeah, yeah.
So as you look ahead to the next 5-10 years in this space, what well, I mean, honestly, the next year or two, right? Like things are changing so fast. But what are some of the things that you think are some predictions or things to watch out for?
Sure. Yeah, I think again we see a lot of modality types within resilience, so we're starting to follow shifts in how we either edit or transduce cells. So moving away from your lentiviral, your retroviral, things like that, more into mRNA or CRISPR even based editing and things like that, for ex vivo therapy. So essentially the cell therapies that you're reintroducing the body. And then beyond that, I think that's the next 5 years. And then going out 5 to 10 years really is being able to do in vivo editing, eventually in the future. So I think those are the two phases that we'll see pop up over the next decade.
Yeah, it feels like at this show we're just starting to hear about in vivo.
Just starting to hear, yeah.
And yeah, there was a mention on stage this morning about someone who's partnering with Moderna on that. But yeah, I mean, one question that comes up with that is like, if in vivo really works, does that make everything else you do kind of like irrelevant? Or
that's a great question. I think it comes down to the price point, really the COGS of therapies as well too. So everybody wants to be able to increase their margins, right, and therefore reduce COGS. So some of these approaches, being able to do it in vivo reduces the higher COGS that you have with cell therapies and things like that. And again, I think we're 10 years away from that, but being able to get to in vivo really reduces the amount of FTE hours that you need, as well as raw material costs. So I think in the future, hopefully that's where we end up.
Not to mention burden on the patient, right?
Yeah, exactly.
It's not an easy thing to go through all the.
Not especially being able to, especially if you have to pull a leukopak for autologous and then re-inject it. It's not the most pleasant experience for a patient, and they're waiting around at that clinical site for kind of the both bookends of that. So you're right. If it's just a singular administration of an in vivo editing therapeutic, it's definitely going to be a better patient experience for sure. Yeah.
Well, thanks so much for chatting with me, Tim. Really appreciate hearing a little bit about what you do.
It's my pleasure, and you had great questions, so it was honestly a pleasure. Yeah. Thank you so much.
Thank you.